Okay, so contagious pathogens are spread or transmitted from person to person. Either inhalation or contact with contaminated items or exposure to bodily fluids, like sex or like Ebola, taking care of patients. That's how most people, it was transmitted in terms of Ebola from patients to healthcare workers and also people who were preparing the body for burial. They'd wash the body and all that, and so basically the whole village would get wiped out. Now, there are some things that are highly contagious, and then, but there's various levels of transmissibility. Things that are contagious in terms of inhalation tend to be the most transmissible. However, they usually are not necessarily all that dangerous. So like influenza and the cold and things like that, most people survive those diseases. And they don't make a person so sick that they actually are less likely to walk around when they're actually shedding viruses. Whereas there's other pathogens that don't rely on contagiousness from one person to another that can be highly virulent, but it doesn't matter that humans are walking around or not, like something like malaria or all sorts of different things, Ebola. So one of the most transmissible diseases is measles, for example. If someone with measles came in here, 90% of the people who were unvaccinated would get it. Okay. Influenza, it's probably 70%, something like that. Something like Ebola, where you actually have to handle or come in contact with bodily fluids and things like that, is much less so. Okay. Now, non-contagious pathogens are still transmissible. They just require some other way of getting it. So, foodborne or waterborne pathogens, like salmonella, like typhoid fever, cholera, those are all transmitted, let's say, through water or food that's contaminated with it. So it's not directly from people, person to person. And some of these diseases, they make you really, really sick, and you're not going to be wandering around with it. You're usually going to be lying there and transmitting it into any food or water that you touch. So like when cholera has been a deadly disease for ages. It's actually fairly easy to survive now. But generally, it dehydrates people. So they end up vomiting and diarrhea, and their guts, they're just passing stuff through it. And that's one of the things the cholera bacteria actually causes. And people die of dehydration. Bacteria are infected by viruses, so it's one of the strongest forces on, in terms of selection, on evolution. And in terms of, we'll get into different aspects of pathogens in terms of how virulent they are and how they're transmissible. Contagious diseases are those that, actually, I want to go, okay. And then there's other things, what we call vector-borne diseases, transmitted usually by animals of some sort. Like ticks or mosquitoes, various other things. The thing about vector-borne diseases is that they don't require people to be walking around. In fact, they're more easily transmitted. If you think about malaria, if you're lying in bed with a high fever, you're not going anywhere, but a mosquito comes along and can bite you. In fact, you're not even going to really pay much attention to a mosquito biting you. And it will bite you. It will pick up the Plasmodium parasite and then go and bite someone else. And it actually, it's kind of interesting. The parasite actually makes the mosquito go and bite someone who's already been infected. I'm not sure exactly the mechanism of that, but it's been shown that instead of biting a random individual, it bites someone who has already been infected, so it's likely to then pick up the Plasmodium and infect someone else. In fact, a lot of pathogens actually control their own transmission by taking over the nervous system of the various vectors. So, a good example is rabies, which you get from an animal who's been infected. It's infected the nervous system. It makes that animal want to go out and bite people or anybody else, anything else that it can get a hold of, and then transmit the disease. Okay. So the question is, what makes some microbes pathogenic? And virulence, or the ability to cause disease, is a heritable trait among pathogens. So if a pathogen is likely, let's say, to not be terribly virulent, it's more likely, let's say, to be transmitted from person to person by being contagious, through inhalation and stuff like that. Whereas vector-borne diseases, like malaria or all sorts of different ones, because there's an intermediate animal that's transmitting the disease from one person to the other, the person can be basically lying there just about ready to expire. And as long as the vector bites that person and then transmit it, it's very simple. And food and waterborne pathogens are, for the same reason, they tend to be rather virulent. So, let's see. So let's take a look at this. On the left, it says more virulent. And here's a patient. He's not walking around. If an insect comes along, so this patient's infected. If an insect comes along, it'll be easier for that insect to actually bite the person because they're just so out of it. So malaria is a good example. So it can be a very virulent disease in terms of pathogenicity, in terms of causing disease, and having strong effect. In fact, often can kill people, like bubonic plague, which was transmitted by fleas from rodents. Now, in terms of less virulent, if you're likely to catch it from someone because, let's say, they're breathing in and out, they're sneezing, all that kind of stuff, that generally requires a person to be still capable of wandering around. Okay? If a person is in bed and not going anywhere, the number of other individuals that he can infect is pretty limited. So a lot of contagious diseases that are easily transmissible are often not that virulent. Okay. So these are ticks. Tick-borne diseases are notorious for being fairly virulent. There's all kinds of different ones. There's Lyme disease. There's Rocky Mountain spotted fever. There's, what is it? A lot of them. So, and then there are diseases that are easily transmitted by food or through the water supply. Typhoid fever, cholera, hepatitis A, food poisoning, that's salmonella. So if we actually look at this, okay, in terms of directly transmitted diseases, on the x-axis, that's down here, right? It's deaths per infection. So at the lower end, very few deaths per infection, and then increasing up to, let's say, 10% or more. So directly transmitted, most of those diseases are rarely fatal. Whereas if they're transmitted indirectly because of, let's say, waterborne or vector-borne diseases, there are some that will not kill you necessarily, but they'll make you pretty sick, and there's others that will kill you. So the way things are transmitted and how sick an individual gets are traits that are selected for in terms of the pathogen. Salute. Now, there's an interplay going on between pathogens and hosts. A host is some individual that can get infected. Some hosts, actually, the primary host will perhaps show no signs of disease. So a good example is rabies. The primary host for rabies are bats. Bats don't actually get sick from the rabies virus. They're the primary hosts. They are, they will, when they're infected though, the virus will cause them to want to go and perhaps bite people or bite other animals and transmit the disease. And so when that happens, then it gets out into the population and it can be transmitted, whether it's to wild animals like foxes and wolves and mountain lions and things like that, or to our pets like dogs and cats. Okay. So there's, as I mentioned, there's different kinds of pathogens. There's eukaryotic pathogens. That means that they are usually single-celled organisms, that the cell has a nucleus and all the organelles. The malarial parasite is an example of that. There's others that are multicellular. A lot of tropical worms are of that kind. So elephantiasis, schistosomiasis, river blindness, all kinds of things. Bacteria, of course, and archaea, and then viruses are the smallest types of pathogens. Okay. So here's some of them. Worms that cause schistosomiasis, trichinosis you get from eating infected pork. Tapeworms, protists that cause, let's say, dysentery, giardia, which is another intestinal parasite. Sleeping sickness and malaria, and then there's some fungal diseases as well that are easily transmitted. So malaria is caused by a parasite called Plasmodium. There's different forms of malaria. There's a less deadly form, it's called Vivax, but the most infectious and virulent is called falciparum. And that's usually what's where malaria is now endemic, and it's basically all over the world in the tropics. That's the form that's being transmitted. It's a vector-borne disease transmitted by Anopheles mosquitoes. There's some anti-malarial drugs that have been developed. So if you ever go to the tropics where it's endemic, it's likely you're going to get, they'll either put you on a drug or they're still trying to develop a vaccine for malaria. That's the Bill and Melinda Gates Foundation. The World Health Organization has really reduced malaria transmission because they've passed around bed netting that has certain insecticides in the material that keeps the mosquitoes away. There's also in villages where it's endemic. Anopheles are always found. Their larvae or their eggs are laid in water, and so people try to get rid of standing water around where they're living. We talked about this already in terms of hemoglobin. So hemoglobin is it's an iron-based protein found in red blood cells that carries oxygen. And we talked about normal hemoglobin that carries oxygen, but those individuals who are homozygous for that are susceptible to malaria. We talked about individuals who are homozygous for the sickle cell allele, and if they're homozygous, they got two copies, and it means that those individuals have very low fitness because they have sickle cell disease. And then there's something called heterozygote advantage or superiority, and those are people who have one copy of the normal allele and one copy of the sickle cell. And they're protected to a certain extent from the malarial parasite. We've talked about all sorts of bacterial pathogens, whether it's bubonic plague, anthrax, E. coli, salmonella, borrelia, it's Lyme disease, all kinds of different pathogens that probably the most common. And then, of course, there's also viruses. And viruses are essentially the ultimate parasite. So bacteria have a lot of their own genes. They have metabolism. They do the kind of metabolic functions that most other organisms do. Viruses, all they have are either RNA or DNA, and then they infect cells, and the cells carry out the functions they need to reproduce. So the viruses actually insert their own RNA or DNA into the genome, and that genome then starts producing the viral particles as well as the protein coats that are around the viruses. And then there's prions, and we'll get to that. So all kinds of different bacteria. Now, I want to make sure you understand, not all microbes cause disease. There's a ton of different kinds of bacteria and funguses and things like that in our gut. They don't cause disease. In fact, it's necessary for proper function and digestion, as well as a lot of other reasons. So we have to make a distinction between disease-causing organisms and those that might very well live on us and in us, that might actually do us good. So bacteria in our intestines, a lot of them produce vitamins like vitamin K and B12. Many human foods, of course, are produced by bacteria and fungi, like milk products. And then a lot of bacteria are actually used, or, well, they're somewhat useful in feeding on oil and petroleum products. So disease-producing bacteria are pathogenic. Now, a lot of the symptoms are because these pathogens release toxins. It might just be their waste products, but those toxins are what are causing the actual symptoms. Some toxins are more dangerous than others, like tetanus or botulism. Now, botulinum toxin, a lot of people use it to get rid of their wrinkles, but it's actually a very deadly toxin that infects the nervous system. So bacteria, as you know, they produce very fast. They reproduce very fast. So here's an uneaten chicken salad sandwich. You leave it out in the sun, come back later, and you eat it because you like bacteria. You decide to eat as much of it as possible, and then you get violently ill. So there may be, bacteria are everywhere. They're on my skin, they're on this table, they're through the air, they're literally everywhere. There are certainly bacteria in the food, probably just a few, but if you put it in an environment that is conducive to bacteria replicating and producing many copies of themselves, then in short order you'll have lots of bacteria. So that's not only shows how fast bacteria can reproduce, and they normally reproduce exact copies of themselves, but there's so many of them that there's also mutations. Most of those mutations probably don't help the bacteria at all, but some might be beneficial, whether it's antibiotic resistance or increases transmissibility between people who are infected. And so bacteria have a whole bunch of mechanisms for actually spreading and being infected, effective pathogens. And because there's so many, there's always evolving mutations. And like I said, most mutations probably don't have much effect, or they have very bad effect on the bacteria, and those don't survive. But some actually are beneficial depending on the environment that they're in. Okay. So bacteria have an extra layer on the outside that eukaryotic cells do not have, and it's called the cell wall. Now, most antibiotics, it turns out, actually degrade the cell wall of bacteria. There's different kinds of bacteria, and their cell walls are made of somewhat different proteins, and so that's why antibiotics work on certain antibiotics, and other antibiotics work on others. All of these are antibiotics, whether it's salmonella, gonorrhea, leprosy, tuberculosis, strep throat, bacterial meningitis. All of these things are bacterial agents. Some are, and they're bacterial pathogens. And so, because they reproduce very quickly, they often make people very sick. And some of them are very hard to get rid of, like tuberculosis. First of all, let's say you have gonorrhea. Usually a 1-week dose of tetracycline or penicillin or something like that will kill it, and you'll be good to go. But with tuberculosis, they have special antibiotics for tuberculosis, and it usually takes 6 months of being on that antibiotic before you can actually clear the infection. So antibiotic resistance is very much like mutations in any other thing. It's like tumor evolution. You have random mutation, you have genetic variation. Just by chance, some variants may be resistant to antibiotics. It doesn't mean that they're completely resistant, but let's say you put in antibiotics and the normal population of the bacteria die, but the resistant form, let's say, is able to live a little bit longer. If you were to stop taking the antibiotics before the infection or the pathogen is completely cleared, the remaining pathogens will probably be the resistant form. And they will then continue to grow and mutate. And just like that video we saw, there will be others that have other mutations that will make it even more resistant, until finally you have a highly resistant form of a bacteria. And that's why a lot of antibiotics are no longer working. And unfortunately, people have been dropping the ball in terms of governments in producing new antibiotics. Now, the drug companies produce the antibiotics that we use. They make money off of it. The trouble is, there's not much money to be made from antibiotics. And so there hasn't been really strong reasons to go and find new antibiotics that work in different fashions from the ones that we've been using for the last 50 years. So before the end of World War II, antibiotics were not available whatsoever. They were discovered first in the 1920s, but they were starting to be mass-produced during the war, World War II, but that was strictly used, limited supply, for soldiers and soldiers who had infections. It wasn't until in the late 40s, early 50s, that antibiotics were finally mass-produced to the point that people were being prescribed antibiotics. And when they were used, they were generally very effective. I recently, during the March for Science, I went down to Pershing Square, downtown Los Angeles, and I met this postdoc at Caltech, and he was working on bacteria. And as I mentioned, usually antibiotics they work most effectively on organisms that are at that point reproducing. And so if an organism is actually reproducing, then those are the ones that are getting killed. Well, some bacteria, like tuberculosis, they go into a quiet phase where they're not reproducing, and they can sit there and they're basically immune to the bacteria, to the antibiotics. And then, because of this quiet stage, they're still there. Let's say you go through the antibiotics, you're finished, but then they start growing again. So that's one of the ways that actually these bacteria it's a mechanism to persist in the environment. So there are some, you've probably heard of, like completely antibiotic resistant bacteria where there's nothing available at this point that will actually kill them. There's a kind of tuberculosis that there are no antibiotics that will kill it now. So in other words, someone who gets those antibiotic resistant, let's say tuberculosis, they're dead. There's no way they're going to survive. It's going to eventually kill them. There's a lot of infections that if you go into the hospital, like staph infections, that are mainly antibiotic resistant bacteria, and usually they have to go with some kind of the last line of defense. Some of those antibiotics are likely to cause, have bad effects on people because they're toxic. And so that's a problem that's really persistent. Our government and governments in Asia and Europe are going to have to start funding ways to get new antibiotics that actually work in a new way. Eventually those might become resistant, but we need something now, or we're back to the kind of the dark ages. When bubonic plague, it's a bacteria. It's actually easily treatable with antibiotics, but if you don't have antibiotics that work anymore, it can cause huge disease. So, when a doctor prescribes antibiotics to you, you have to go through the whole series. They say, take it for a week, and you only take it for 2 days because after 2 days you're feeling better. It's likely that some of the bacteria are still alive. They might be variants that have slightly more resistance than the normal kind. And so after 2 days, you stop taking the antibiotics and they start growing and mutating again. Now, they used to hand out antibiotics like candy because they didn't think about the evolution in terms of the evolution of pathogens and bacteria. And bacteria, of course, evolved just like anything else. So doctors would hand out this stuff and people would take it, but maybe they wouldn't take it very well, or maybe they had a virus and an antibiotic won't work against a virus whatsoever. There's another thing that they're doing. If you give, let's say, chickens or cattle antibiotics like Cipro or Levaquin, they grow faster. So a lot of the industry that's producing our meat feeds their animals antibiotics. Now, since they're feeding it, all the animals always have antibiotics in them. It's likely that eventually there will be some that evolve resistance. And then you've got an antibiotic-resistant bacteria, and we might get it from the food we eat. Plus, the food we eat still has some antibiotics in it. So, of course, there's an easy way to get around that, and that's having a more humane way of growing, let's say, chickens. So now, normally what they do is they take chickens, put them in these little tiny cages. They can't move whatsoever. They feed them, they grow, and then they slaughter them. With such a high concentration of chickens and chicken waste, it's a perfect ground for a medium for creating bacteria to grow. Now, if you had a different system where the chickens were more spread out, there'd be less likely that they would pass infections back amongst themselves. But because they can use antibiotics, they can put these chickens in these really kind of dangerous situations. So I know in Europe, there's a lot of regulations now that they can't make those kinds of chicken farms anymore that you might see in Arkansas or whatever, with basically hundreds of thousands of birds in these chicken farms. Now, the reason, by the way, they're able to do that is because these companies that produce chickens or cattle or whatever, they have a lot of sway. They have lobbyists that lobby congressmen, and congressmen are not notorious for knowing very much. And a lot of them don't understand science. Okay, now, viruses. As I mentioned, a virus does not do any metabolic functions by itself. In fact, some people don't even consider viruses actually living, but frankly, I do. They're just the perfect parasite. Now, most parasites, what they do is they get inside, let's say a tapeworm, they get inside the body, and they feed on the food that you eat, and they grow bigger and bigger, and they release eggs out into the environment, and that's how they're passed on from one person to the next. So a parasite lives inside or on a person or some other organism, and is basically getting everything it needs from that host. Viruses are, in a sense, a perfect pathogen because they don't even have cells and metabolic costs. All they do is they infect cells, insert their genetic material into those cells to make copies of themselves. They're exceedingly common. There's some viruses that are fairly innocuous, like the cold virus. There's others, of course, that are deadly, like Ebola or HIV, just like with bacteria. And they have all the characteristics in terms of being transmitted. They can be transmitted by everything from just contagious stuff between one individual and another, or vector-borne viruses, or waterborne viruses, all those kinds of ways of transmitting. So these are all electron micrographs of viruses. Because they're very small, most of them you cannot see under a light microscope. The light microscope can only magnify up to a certain point, and beyond that point, it can't. So instead of using photons, we use electrons, and you can see things that are much smaller. So rabies virus, tobacco mosaic virus, in other words, viruses infect plants as well as animals. Influenza viruses, the bacteriophage is actually a virus that infects bacteria. They recently discovered some viruses that are actually huge. And there's a great debate about whether they actually constitute a different domain of life or whether they actually gave rise to most bacteria that are strictly parasitic. So as I mentioned, viruses hijack the host cell to produce new viral components. Okay, so here's a virus. That's the genetic material in there, and then on the outside is this protein coat. In this case, this is a virus that uses RNA instead of DNA. They're called retroviruses. So retroviruses use RNA instead of DNA. And they use RNA reverse transcriptase to take that RNA and turn it into DNA. The DNA is inserted into the genome. It's transcribed, translated in the host cell, and then that DNA produces all the components of the virus, including more RNA, as well as usually the protein coat of the virus. And the protein coat is important because that's how the virus actually infects cells. The proteins interact with certain cellular proteins on the outside of the cells to actually infect the host. So that's basically how viruses work. A lot of viruses are cell-specific, also species-specific, whereas others can actually infect a lot of different organisms at once. Rabies viruses can infect a lot of mammals as well as people, and they actually have a primary host in bats, and the bats actually don't get sick. The HIV viruses attack our immune system cells, the T cells and the beta cells. And that's why people end up with an immune system that doesn't work anymore, and they get all these other diseases that from pathogens that normally would not cause disease in humans with a regular immune system. I mentioned this earlier, a disease that's transmitted from one organism to another, like between an animal and humans, is called a zoonotic disease. Okay. In fact, influenza is a zoonotic disease. The primary hosts are actually birds, and birds do get sick and die. And then here's a bacteria, and all around it are these phages, are viruses that infect the bacteria. As I mentioned earlier, some bacteria, a lot of bacteria have actually evolved defenses that recognize the DNA or RNA from a virus and then have enzymes to cut that virus up. Some viruses, like I said, they have RNA, they're retroviruses. Others have DNA. So you can have both kinds of viruses. Some viruses are linked to cancer, which is something you would expect because if you're taking foreign DNA and inserting it into your genome, that can act like a mutation and cause cells to start behaving uncontrollably. So HPV and cervical cancer and throat cancer are caused by a virus. Most people who get infected with it, actually their immune system clears the virus, but some people don't. Some viruses are can become latent. In other words, they're not in their disease-causing phase. a good example is chicken pox and shingles. It's the same virus. So after you get chicken pox, you're usually immune to it, but the virus has now migrated up and is hanging out in your neurons. And it might stay there for the rest of your life and nothing will change. It won't cause any disease or anything like that. In fact, we have a whole bunch of viruses that don't cause disease in our genomes, just hanging out. Their DNA or RNA is hanging out there. But as people get older, their immune systems don't work as well, and so sometimes they get shingles. Or, let's say they've got cancer, they're on immunosuppressive drugs, or their immune system doesn't work, and you get shingles and they migrate down from the neurons and then start causing symptoms. So, like HIV is a great example where the HIV virus can actually hang out in the cells, various different kinds of cells, not cause disease. So when someone is being treated for HIV, they have a 3-drug cocktail, and it keeps the virus at a very, very low, in fact, it's not detectable in the bloodstream. But it's still there inside certain cells, the DNA of the virus, or the RNA of the virus. If you were to stop those drugs, they would reemerge. So those drugs actually don't cure HIV, they just manage it. And they use, by the way, they use 3 different cocktails because each drug works on a different part of the life cycle of the virus. So originally they only had 1 drug for HIV. It was called AZT, worked, but then the virus evolved defenses and it no longer worked. So with 3 different drugs working on 3 different mechanisms, it's unlikely that the virus will actually evolve resistance to all 3. Okay, so HIV, AIDS. It primarily infects T cells, the part of the immune system, but it also turns out HIV infects a lot of other cells. Like in the spleen, a lot of the cells are stem cells, and it will infect those. And in fact, HIV, inside a body, you often find various different strains in different kinds of tissue. Now, most patients, they die not from the HIV virus, but from infections that our immune system would normally protect us against. HIV transmission, it's a hard thing to transmit. You can only do it either by having sex or by using drugs, injectable drugs with dirty needles. Or, unfortunately, let's say transplants or blood transfusions. That's why all those things have to be tested before they're used. You can't get HIV by kissing someone. Now, when HIV first showed up, people were just terrified of it, of course. It was a virtual death sentence. But let's see, what was I thinking? So, okay, so it was a virtual death sentence, but it's not now. But, oh, I know. In fact, since it can be transmitted through the blood, right, a lot of people thought, well, maybe it can be transmitted by mosquitoes. And there was a big, mass panic that HIV, people with HIV could transmit it by mosquitoes. And there were all these laws to be put in place to quarantine people with HIV. We don't want this HIV-infected boy or girl in our school because they were afraid. It turns out that you can't get HIV from like a mosquito or a vector-borne disease. It requires close transmission and contagious, and usually by sex. RNA viruses, retroviruses, because they have RNA, it's easier for mutations to occur in RNA. If you remember, DNA is double-stranded. So if you have a mutation, it can be corrected because there's a complement to it. And there's a way for the body, they use certain proteins to make sure that if there's any sort of mutation, they're corrected. But RNA isn't like that. So RNA viruses tend to be highly mutagenic and have a high error rate, and that's how you end up with lots of mutations. And like I said, mutations, most are bad, but some help the pathogen to actually infect and carry on the transmission. So just like I mentioned, HIV can hang out in different kind of cell types. Each of those different cell types might be a better environment for a certain strain of HIV. So as someone is infected with HIV, it tends to evolve inside their body and having different strains and different kinds of tissue. Okay. I think we'll end there.